As we have discussed in the last video about principle of biochemistry, we will continue about the basic knowledge of cell organelles in this video. Now we will discuss about chloroplasts. So chloroplasts are green plastids and they are the organelles found only in plant. They are not found in animal cell, and it is a eukaryotic and also that conducts photosynthesis. So they are the disc shape, which it is shown here, and it is about 5 to 8 micrometer in diameter. And it is 2 to 4 micrometer thick. And a typical plant cell has 20 to 40 of them. Chloroplasts are green because they contain chlorophyll and the pigment that harvests the light used in photosynthesis. Also, this captures light energy to conserve free energy in the form of ATP and reduce NADP to NADPH through a complex set of processes called photosynthesis. The direct reaction of photosynthesis also takes place in the chloroplast. So, inside the chloroplast, the material which is present, it is called as stroma. So, inside this we can say it is a stroma. And also within a stroma there are stacks of thylakoids, which is here it is this single unit, it is called as thylakoids. And these stalks of thylakoids, stalks of thylakoids, it is called as grana. And thylakoid have a flattened disc shape, which you can see it is a round and flattened disc shape, and inside it is an empty area called thylakoid space or lumen, which is we can see this is an empty area, we can say it is a lumen or thylakoid space, and a photosynthesis takes place in this thylakoid membrane. And the next is cell wall. So cell wall gives a definite shapes and provide protection to the protoplasm. So in this diagram you can see this is cell wall, this is cell wall, this is cell wall, and this is a particular cell, and in this is cell, or this is all this cell wall. And the cell wall is usually a non-living in nature and it is permeable. Also, the cell wall, it is made up of fibrils of cellulose in plant cell. And this cellulose is embedded in a matrix of several other kinds of polymer such as pectin and lignin. It also provides a mechanical strength to the cell, and the cell wall consists of usually 3 parts. So first of all, it is a primary wall. This is a primary wall, and it is usually made up of cellulose. And this is the secondary wall, and it is also called as a dead cell, and tracheas and sclerenchyma as an example. We can say it is present in plant cell only, and this middle part, it is called as middle lamella. And this middle lamella, it is made up of pectic acid in the form of calcium and magnesium salt. So the another organelle is a ribosome. Ribosome, it is a small organelles, and usually there are the large subunits and small subunits. And ribosome is also a protein synthesis site, and it is composed of RNA-rich cytoplasmic granules. And also in ribosome, RNA is present in 65% and the protein is present in 35%. So the next is lysosome, and lysosome, it is a round organelles and surrounded by a membrane, and contains a digestive enzyme which, and its function is to digest foreign particles which are brought inside the cell during the phagocytosis and pinocytosis, and it also serve to digest cell components after cell death. So this is a simple diagram of Golgi apparatus, and the Golgi apparatus consists of a flattened single membrane vesicle. So here we can see in this diagram, this is a single membrane vesicle and a flattened type, and some become vacuoles in which secretory products are concentrated. And the primary function of the Golgi apparatus is to process and pack the macromolecules such as proteins and lipid. This is a main function and also a primary function of Golgi apparatus. So it is important in the processing of protein for secretion. So that's why it needs a processing. Next is vacuoles. So vacuole is a membrane-bound organelle which is present in all plant and fungal cell and also in some animal cell. Usually in animal cell vacuoles is not present, but in plant cell it is present in very big size. We can say that half of the cell is half of the cell space is occupied usually by the vacuum. So the vacuum is surrounded by a tonoplast, which we can see in this diagram. It is a tonoplast, and they are essentially enclosed compartments which are filled with water containing inorganic and organic molecules including enzyme. So the function of vacuoles are, first of all, the function, it is it helps in isolation of a material that might be harmful or threaten to the cell, and also some waste product which needs to be removed from the cell. It also function for that too. So the functions of vacuoles are, it helps in isolation of materials that might be harmful or threaten to this cell. Also it helps in removal of waste, and also it maintains the hydrostatic pressure or turgor in the cell, and also helps in maintaining its internal pH. That is it for today. We will see you in the next video.